So this is the tyrosine, and it has several unique things that you're going to remember about it. One is that when it gets a ligand, it's going to dimerize. So di means two, okay? It's going to dimerize. There's going to be two ligands, and there's going to be two receptors, and they're going to hook together. And if you see on the one, it actually shows that they're going to hook together with some sulfide bonds. For the chemistry people, you'll see that means they're hooking together. The unique thing about these is that they autophosphorylate. So they phosphorylate each other, okay? They don't have to wait for any other proteins to come passing by. They can just do it for each other, okay? Autophosphorylation. So this is the unique thing about them, is that they don't necessarily need help from the outside. They don't need to have a bunch of other, proteins come hang out by them and give them and activate them. The other thing is they're involved in many forms of cancer, like I said, because of the fact that they are involved in the growth and reproduction of the cells. Okay? Okay. So this is the EGFR, which is epidermal growth factor, and you can see that it has a combination of two things going on. It is also attached to the G protein, okay? So that means that we're going to have two things happen. On the one hand, it's going to phosphorylate itself, and on the other hand, the G protein is going to stop the phosphorylation. You see that little line on the end of there? That always means stop, okay? If you see an arrow, that means it's moving forward. If you see a line, that means it's stop. So this is a control mechanism, okay? It's a control mechanism to make sure that there isn't too much activity on this receptor, okay? Okay. This is the story, which you can read later, and I'll just kind of tell you about it. So there is, like I said, every time somebody finds one of these things, they get to name it for whatever they want. And sometimes what happens is that somebody has studied one kind of tissue and another person studied another kind of tissue, and it turns out that they have just named something twice and it's really the same thing. Okay? So then depends on, who's more famous, who has more papers out, or, then what you end up calling this, okay? But usually there's a while there where there's two names going on and depends on which field you're in, which one it's going to be. So in this case, it's called the ERB protein family of epidermal growth factors. Okay, so that is one of the names that it was given. And it's been involved in multiple sclerosis. It's been involved in Alzheimer's disease. Your endothelial cells are one of the most common forms of cancer, whether that is inside your lungs, whether that is breast tissue, whether that is brain tissue. It all derives from endothelial or epithelial cells, okay? So that's where these all come from, okay? So they found out that there is excess ERB signaling in cancer, okay? That there's too much, that it's making too many cells, okay? That it's the signal that says keep going, keep dividing, keep reproducing, okay? So they came out with this great drug, which is called Herceptin, because Herceptin is actually for the HER2 receptor, except that the HER2 receptor is exactly the same as the ERB2 receptor, okay? So when they were studying breast cancer, they found this HER2 receptor that was upregulated in some forms of breast cancer, and they came up with this cool drug that would block the receptor. The only problem is that since the HER2 receptor is exactly the same as the ERB receptor, and there's other parts of your body that have the ERB receptor, now you have a problem because you're not just stopping the place where there's extra growth, okay? As it happens, ERB2 is also really important in your heart. And so unfortunately, when they give you the Herceptin drug, they have to be very careful that they don't cause heart damage, okay? Because there were two receptors that were exactly the same, but they were discovered in separate tissues, and it took a while before they figured out that it was all the same thing, okay? So that's kind of one of those stories of sometimes medicine does unexpected things because it's such a big area of discovery, and we don't know the whole story. Okay, insulin. Most people understand insulin. They know it has something to do with sugar. They may not know how the whole thing works together, but they know it has something to do with sugar. And insulin is actually a ligand, so to speak, for a receptor tyrosine kinase. Okay? Now, like I said, it wants to dimerize. Let me see if I can actually make this work. There. Okay. Oh, that's going to be better or not. Okay. So we have the receptor, and it wants to dimerize. The insulin actually looks like this and sits on top of there, and this is the cell membrane. Now, what insulin's job is insulin says that glucose is coming, okay? The insulin doesn't actually carry the glucose. It's just the messenger that says the insulin is coming. So this receptor has a job, and its job is to recruit a transporter to take the insulin into the cell. So the point of this receptor is to then come down and take these vesicles that have got glucose receptors in them and get them to go join it up there at the cell. Okay? So that's how the insulin works. In type 2 diabetes, which is the kind of diabetes that you usually see in older people, there is what's called insulin resistance. So what happens is this insulin comes to sit on this receptor and says, there's glucose out here, we want to put it in the cell. So, move out those transporters, we want that insulin to go in your cell. And the receptor says, we don't need any more glucose in the cell, we've got all we want. You're overfeeding us, we're getting fat, we don't want that insulin. We don't want that glucose in our cell. And so what happens is your body puts out more insulin, and more insulin, and more insulin, and more insulin. And then the cell says, all right, we'll take it. So then all the transporters come to the cell membrane, and they gobble up the glucose and take the glucose into the cell. And then, because of the fact that there was so much insulin made, and there were so many signals that said, take me in, now you have low blood sugar. Okay? So now there's no sugar left in the blood, and the only sugar that's going to make it to the brain is going to come out of the blood. So you've got low blood sugar, and that means your brain is starving for the sugar. So that's when you get shaky. That's when you sweat profusely. That's when your hands are like this and you have kind of confusion in your brain. Oftentimes you feel very weak, and that is because you have got all that sugar, all that glucose has finally entered the cell. Okay? So it's a bad cycle because you eat, you have the sugar out there, then you have too much insulin, and then you have not enough sugar. Okay? So this is the beginning of what happens with type 2 diabetes. Then what happens is that you have made insulin so much that your beta cells, which is where the insulin is made, they get tired and they wear out and they literally quit making it at all. Okay? So that's the tyrosine kinase, and that's its connection to the insulin. Okay. So we got one more movie. And then I think we got a couple more slides after the movie. Okay, so this one says Introduction to Cancer Biology, and it's going to talk about the signaling pathways that have to do.
An Introduction to Cancer Biology. In this presentation, we will discuss selected characteristics common to many cancers. Abnormal signal transduction resulting in uncontrolled cell proliferation. Loss of apoptosis or programmed cell death. Tissue invasion and metastasis, permitting spread of the cancer. And angiogenesis, leading to enhanced blood supply of tumors. Normal cells require signals, usually delivered by ligands, to stimulate their growth and to tell them when to stop growing. These signals, or ligands, can be in the form of growth factors and inhibitors, extracellular matrix components, or cell adhesion molecules. These signals are transmitted into the cell through proteins found on the surface of cells called receptors. Each ligand binds to its own specific receptor. Receptors often consist of three domains: an extracellular ligand binding domain, a transmembrane domain, and an intracellular domain. Binding of a ligand to the extracellular domain activates the receptor tyrosine kinase, which activates other proteins by phosphorylation of, adding a phosphate to, the amino acid tyrosine on a protein inside the cell. When a ligand binds to the receptor, a signal goes to the intracellular domain, activating the associated enzyme and initiating a cascade of signals to the nucleus that tell the cell to grow and divide or to stop growing. Malignant cells generate many of their own growth signals, which allows them to divide with reduced external growth stimulation. Some cells are able to produce their own growth factors and stimulate their own growth. This is called autocrine stimulation. For example, glioblastomas express platelet-derived growth factor, or PDGF. Sarcomas express tumor growth factor alpha, or TGF-alpha, as well as epidermal growth factor receptor, or EGFR. In normal cells, the production of cell surface receptors is limited by cellular restraints on gene expression and protein translation. In tumor cells, however, mutations in the genes encoding for the receptors disrupts this finely tuned regulation, and too many copies of the gene are produced, a phenomenon called gene amplification. This, in turn, leads to excessive transcription and production of receptors. The end result is a higher than normal number of copies known as overexpression of the receptor on the tumor cell surface. This gives the tumor cell increased potential to be triggered into a growth phase by the binding of ligands to the excess receptors. The more receptors expressed, the more binding sites are available for the ligands. Gross overexpression of growth factor receptors can result in ligand-independent signaling, where receptors are active in the absence of stimulating molecules. Structural changes to a receptor can also lead to ligand-independent activation. For example, truncated versions of the epidermal growth factor receptor, where much of the intracellular domain is missing, are constitutively active. EGF receptor, also known as HER1 or ERB1, is a member of a subfamily of type 1 receptor tyrosine kinases. These receptors are found primarily in the membranes of normal epithelial cells from skin, breast, colon, and lung, amongst others. EGF receptor and its ligands play an essential role in the regulation of cell proliferation, differentiation, and survival. EGFR is overexpressed in tumors arising from the colon, rectum, and head and neck, to name a few. When a specific ligand binds to its receptor, this leads to changes in the receptor that transmit a specific signal into the cell. For example, the receptor tyrosine kinase is activated and initiates a signaling pathway specific to that receptor. This phenomenon is called signal transduction. Activation of a signal transduction pathway creates a complex chain of events in the cytoplasm or fluid intracellular space that eventually leads into the cell nucleus, where the transcription of genes regulating cell cycle progression are stimulated, resulting in cell proliferation. One of the major cascades implicated in cancers is the Ras-Raf mitogen activated protein, or MAP, kinase pathway. Another interesting pathway is the phosphosinositide 3 kinase, or PI3K AKT mammalian target of rapamycin, or mTOR pathway. These pathways are linked to each other and other signal transduction pathways in the cell. Deregulation or loss of normal controls in any of these pathways is thought to be present in all human tumors. Once the signal reaches the nucleus, transcription factors are activated. These factors transcribe the genes that are translated into the proteins, such as growth factors, that are necessary to allow the cell to continue to proliferate. The end result of any growth factor receptor signal transduction pathway is tumor DNA replication and cell division. One tumor cell becomes 2. If mitosis continues, 2 tumor cells become 4, with exponential growth potential. On the tissue level, this leads to increased tumor growth and increased tumor size. Many of the components of these signal transduction pathways are potential targets of anti-cancer therapies. Therapies can target the ligands, receptors, intracellular second messengers, and nuclear transcription factors responsible for tumor growth. Ligands can be neutralized before they bind to the receptors. An example of this is bevacizumab or Avastin, which is a humanized monoclonal antibody that targets circulating vascular endothelial growth factor or VEGF. Platelet-derived growth factor or PDGF and fibroblast growth factor or FGF are other examples of ligands that can be targeted for different cells in the body. The receptors on the surface of normal and tumor cells can be inhibited directly. Cetuximab or Erbitux is an example of this. It is a chimeric antibody that binds directly to the epidermal growth factor receptor and competitively inhibits the binding of EGF and other ligands, such as TGF-alpha. Another way to block the receptor's function is through small molecule inhibitors of receptor phosphorylation associated with them. For example, EGF receptors have a tyrosine kinase that can be blocked by the molecules gefitinib or Iressa and erlotinib or Tarceva.
Okay. No, I don't expect you to remember. I don't expect you to remember all those names of those cancer drugs, but you kind of get the idea of where the targets are, of what they're trying to design, or how they're trying to combat cancer. And it gives you an idea of just how important the receptors are, for, not only for our normal function, but also when there is misfunction. Okay, we're going to talk about just a couple more kinds of receptors, although I think you're getting the idea now that they have a lot in common, each one. Guanylate cyclase is a special kind of receptor. The most common one is involved with atrial natriuretic peptide. When we get to the heart, we will talk about this. But atrial natriuretic peptide is basically the primary thing that is going to keep you from going into congestive heart failure. It actually is a sensor that sits in the atria of the heart, and if there's too much fluid in the heart, it senses the stretch and it senses that, and it'll send out signals for you to get rid of some of the fluid that's in your body. The unique thing about this one is it does the cyclic GMP cycle, which is different from the ones that we have seen before in terms of its cascade. The other thing that is interesting about this, and they just mentioned it a little bit in the movie, is that there's actually 3 of these receptors. They have an A, B, and a C. One of them has nothing on the inside. So the domain that's inside the cell is gone. So basically all it does is capture the ligand, and that's just a clearing mechanism. It's a way of cleaning up extra stuff in the cell. And that's what the guy was mentioning, that some of them don't have anything on the inside. That's because they're just there to catch the ligand on the outside and clean it up because they don't want any more activity. Okay? JAK-STAT is another one of those ones where somebody got really wild when they were doing the naming. So this is called the JAK-STAT pathway. Again, it's a tyrosine kinase. It also makes a dimer, and there's got to be two of them together. And the JAK and the STAT just belong to right here where it's called Janus kinase. So you know a kinase means it's going to phosphorylate. And STAT means signal transducers and activators of transcription. So this one is specifically going to be involved with going to the nucleus, and usually it helps unroll the DNA so that it can be transcribed. Okay. This is a different pathway than we have talked about in the past, and it's very unique, and it has a lot to do with you and your body and the drugs you take every day. This is called the arachidonic acid pathway. So what's an arachnid? Anybody recognize that word? Spiders. Okay? So they found out that this is the same kind of a molecule that's in spiders. Okay? There's a lot of toxins that are used chemically that come from spiders, and this happens to be how this one started. So arachidonic acid is where we start. The parts of it that you are going to be very familiar with is over here on this side where it says COX. Okay? COX is also known as cyclooxygenase. Okay? There's a whole group of drugs called non-steroidal anti-inflammatory drugs. That means Aleve, ibuprofen, Motrin, Celebrex, naproxen. Any of these names sound familiar to you? They're used all the time because they're anti-inflammatory. They also are antipyretic, which means they're going to reduce fever, okay? And they're analgesic, which means that they take care of pain. So somebody may use it if they are an athlete and they sprain their ankle or they're having a knee problem. You may use it if you are a pianist and you've got really sore fingers, okay? You're going to be using it if you have got high fever. You may be using it if you have very bad headaches, okay? These drugs interfere with that molecule right there that says COX, okay? Right there. So there is a large group of drugs. They're also used for rheumatoid arthritis and for osteoarthritis. And their whole point is to reduce inflammation. That's the primary thing, okay? So if you take a crash on your scooter and your knee is swollen, that's what you want to take, okay? Ibuprofen, one of the most commonly prescribed drugs. It used to be all prescription, but it is over the counter now, okay? Okay. Does anybody recognize the word turmeric? Raise your hand if you know what turmeric is. You might also know it as curcumin. You know what curcumin is? Okay. You may also know it as kunik or kuminchan. Okay. Okay. So turmeric is a spice. The active ingredient inside the spice is called curcumin. Okay? Curcumin is given all the time. This is what you would call a folk medicine. And I don't mean that in any way disrespectful. I just mean it simply that it's something that's been around a long time. Okay? So, how many people in this classroom got turmeric in milk when they were sick when they were a kid? Nobody? Your moms all got past that? Nobody got turmeric stirred into milk when they were a kid? Okay. Turmeric is actually a very good compound, and thanks to cell signaling and to a lot of work by a lot of people, we actually know how it works now. Okay? So this is one of those folk medicines that we found out really does do a good job. Okay? And one of the things that it can do is that it can help when there's been cell damage. Okay? It is given to cancer patients to help them repair cell damage to their normal cells. Okay? To their normal cells. And it actually goes, I think, through a tyrosine kinase, if I remember right. I think that's a tyrosine kinase. It actually goes through a tyrosine kinase, and it causes repair within the cell so that the cell won't die. Okay? So sometimes the folk medicine actually turns out, when we figure out how it works, we can say, yes, you already knew it was good, but now we can tell why it's good. Okay? This is one of the biggest breakthroughs in medicine that's been around for about the last 10 years. This is a whole new group of drugs that were basically invented to help people who have rheumatoid arthritis. So rheumatoid arthritis is the one that is highly inflammable. It's not old age wear and tear. It's usually the one that young women get, where they have very, very inflamed joints, and the joints actually get destroyed from the inflammation. It used to be that the only thing they could really do was to give them those anti-inflammatories that I talked about the first time. But they've actually developed an entire group of drugs that work through this pathway, the TNF pathway, in order to stop the inflammation. As a group, they call these drugs the biologicals. Okay? And these drugs, Remicade, Enbrel, some of these drugs are probably seen on television because they advertise a lot, but they're all very, very effective in stopping the inflammation in rheumatoid arthritis. And the reason that those were able to be developed is because of the fact that they first figured out what the pathway was that was causing the inflammation, okay, which is the TNF-alpha pathway. Okay. Now it's time to draw. I want everybody to get out your paper. And you are going to make your own diagram of how this is supposed to look so that it will be committed to your memory forever. Okay. So make yourself a cell membrane. Make yourself a receptor and a ligand on the surface of the cell. Okay, make yourself a G protein with an alpha, beta, and a gamma. Now you remember what I told you about the beta and the gamma staying together and staying close to the membrane. So we're going to bring our beta gamma over here. Should actually be 2. Okay, we're going to bring it over here and we're going to attach it to the PLC. And we're going to get our alpha subunit to come down here. And we'll make this an alpha S just to make it easy, okay? And our alpha S is going to attach to adenylene cyclase. Okay, and then that means that adenylene cyclase is going to make cyclic AMP. Now, if you have a lot of colors, you could do this in color. It might be easier for you. And the cyclic AMP is going to make pKa. You can make them different shapes if you want. pKa is going to go phosphorylate something else, and it's going to eventually go to make a message going to the nucleus. So you can stop at the pKa, okay? Over here with our beta and our gamma and the PLC, we are going to end up with DAG and an IP2, which is really going to turn into an inositol triphosphate, I believe. IP3, right? IP3. Okay. That's a 3. Okay? The DAG is going to go and phosphorylate PKC, and the IP3 is going to come down and release calcium from the ER. Okay. Remember, I told you that I flunked kindergarten art, so I'm sure yours looks better than mine. Okay. Now I know if you did this and you actually drew it out, that means that you are going to know this. It's going to be like in your brain forever, and you'll be all ready if it's on a test, right? So usually this is the kind of stuff where I put it on a test, and then it'll be worth a certain number of points, and I'll be looking for certain things. So example, I'll give you a point for drawing your receptor. I'm going to give you a point for your AB gamma, and I'm going to give you a point for separating it. And I'm going to give you a point for each one of these things that's supposed to be here. Okay? So this would be considered, quote unquote, a short answer question, and it may be worth 10 points. Okay? Does that make sense? Okay. Everybody think they got the idea? Okay. Okay. That is it for today. I'll see you next Thursday.